Assalamu alaikum everyone. Can you hear me? Can you see the screen? Please answer in the chat. Okay, thank you. My name is Hafss Bahsaiwth. Today, inshallah, we're going to be explaining the RNA, the second lecture, the RNA processing. If you have any question, just go ahead and put it in the chat. I'm using my phone. I'm using the audio from my phone, so sometimes I might not be able to see your chat, but I'll try to check from time to time. Okay. Okay, we can start. The RNA processing. The slides, some of them are mine, some of them I took from the RNA second lecture by Dr. Ahmed Al-Jada. Okay, so just let's start. This is just a general checkup on your vocabulary. I expect you guys to know some of these vocabulary given in the previous lectures. So this is just a cell. You can see just a cell. This is the nucleus. Hold on a second, please. Yes. Okay, so this is a cell. This is the nucleus. You can see you have in DNA. This is the cytoplasm. Don't worry about all of this. This is just a vocabulary or terminology checkup. DNA, you have the DNA, unwind the DNA. Okay, you have the messenger RNA here. It's being formed. After it's being formed in a process called the transcription, you're gonna exit the nucleus, go to the cytoplasm, and go to something called the ribosomes. The ribosomes, in a process that's called the translation, gonna make the proteins using the help of something called the transfer RNA. It's gonna help them build the protein. This is almost the same example, having a cell, a nucleus over here. You have DNA that's being transcribed to messenger RNA. There is a lot going on between here. We're gonna dig into that in the next slide. So DNA transcription is gonna go to mRNA. mRNA will exit the cell, go to the ribosome, and in a process called the translation, we're gonna have the proteins or shorter form of the protein called polypeptide. Gonna check a little bit about the DNA. What's the DNA exactly? DNA, just have a look. Don't worry about all these terminology. Just focus on what I'm saying. I'm trying to focus on just terminology. You guys understand some of these terminology. You have, this is one strand of the DNA. DNA is two strands. This is just one strand. You have the enhancer and you have the promoter region of these. The enhancer, you can think of it as like a road sign that tells you like, okay, exit 19 is after like 15 or like 15 meters or something like this. So it's at a distance from the starting of the transcription location. The RNA polymerase is going to come, get attached to the promoter, and start the transcription. Back to the process, you get the DNA transcribed to the mRNA. Here the mRNA, but usually there is something before the mRNA. there's something called the pre-mRNA. It's like the raw mRNA. It's unprocessed. We did not change anything. We just have it transcribed, and we're going to have some processing for it, making the mRNA. What's happening here is called RNA processing. This is our lecture. We did not start our lecture. If you have any questions, just write it in the chat, and I'll check regularly. Okay, so this is the lecture. RNA processing is the process that goes on, like what's really going on here after the transcription, but before we having a mature mRNA, what's really going on here? There are some changes that are applied to the mRNA until we have the mature, complete mature mRNA. The process usually have 4 steps. The first step is adding a cap at certain end. So basically, we have this strand of messenger RNA. We're gonna have a cap and a tail and some other processes as well in the middle here. Don't worry, we'll get to them. So we'll start by adding the cap. This is a protein. Before we add the cap, this is the messenger RNA, the pre-messenger RNA, and this is the mature. This is the pre-RNA, and this is the mature, full mature RNA after the process. What's happening here at the middle, there are certain processes that are going to be applied to change the messenger RNA to the pre-messenger RNA to the messenger RNA. All of this yeah, so all of this, starting from here, including the pre-messenger RNA and the processes, all of them are called heterogeneous nuclear RNA. Guys, you have to focus. It's called heterogeneous nuclear RNA, both included. It's really important concept. So the heterogeneous nuclear RNA is going to include the pre-messenger RNA and the processes that happened before. We call it mature mRNA. What's the processes? The processes, the first one, we will call it adding a cap. Adding a cap. What is the cap? This is the cap, you can see it. Pre-messenger RNA, the first addition, the first process is adding a cap of something. What is that? It's called methyl guanosine cap at the 5' end. So this is the pre-messenger RNA. Guys, am I going too slow, too fast for you? Let me know in the chat. Do you want me to go faster, slower? A bit faster, sure. As long as you understand, I'll go faster. So this is the pre-messenger RNA. We have it here. Just follow the pointer if you can see it. We will add a cap at the 5' end or the 5' end. So this is a 5' end, and this is a 3' end of the messenger RNA. The cap, it comprises of the 7-methyl guanosine cap at the 5' end. There are too many names for the 5' end. Another name is the amino group, and another name for that as well as the formula of the amino group, which is NH2. So it has many names: the messenger RNA 5' end, the amino group, and and NH2 end. It has a cap of a 7-methyl-guanosine cap. You have to memorize that, and it's in the 5 end. A way of remembering this, I would use like a symbolic representation. I would use like a cap. I'll write in it like 7 mg, like methyl guanosine, and the cap is on the 5 prime, or that's called the amino group end. Another mnemonic also I would use myself to memorize this, something like what is the 7 mg? Like I can call it the 7th medical guide, the 7th medical guru, whatever you want to call it. If you have a better mnemonic, then you can use that. This is just my way of remembering some of these stuff. So a 7 mg or medical guide cap gonna be put on the 5 prime end, or the amino group end. So first process is the cap. What's the function of the cap though? The function of the cap, the first function, there are 2 functions. The first function is to stabilize the messenger RNA. This one, the messenger RNA or DNA in general, is actually fragile or volatile. It can be degraded, so you have to stabilize it. So this one, the cap, is actually going to stabilize it and prevent the degradation. It's also going to serve as a binding site. Remember, when the messenger RNA, when it goes outside the nucleus, goes to the ribosomes in order for it to get translated. So this is the binding site to start the process of translation into a protein. So we're done with the cap. The next thing is going to be a tail. So we added a cap here. The tail is going to be added here. But hold on a second. There is something before the tail. You have to do something before you add the tail. So the first step, as you can recognize here, there is something called the poly-A addition signal. There is a signal that tells the polymerase where exactly to add the poly-A tail. So the first step is actually an endonuclease that's going to come. That's an enzyme. It's going to come to this poly-A addition signal, and it's going to cut some part of it. After that, we will add the second step. We will add the tail. The tail comprises of 200 A's or adenylates. So we will add around 200 here of adenylates. We'll attach them after the nuclease came here and cut part of the signal. The signal, it has a specific structure. The signal is AAA, U, is AA, U, then AAA. So first step, it's going to cut the part, then attach the tail that has 200 A's or adenylates, too many of them. What's the function of the tail? Or before that, the addition of the tail is going to be in the 3 prime end. Another name for the 3 prime end is the carboxyl group end, or the chemical formula for the carboxyl group, the COOH. What is the function of it? One of them is also to stabilize, the same as the cap, to stabilize the mRNA, prevent degradation. The second one is that it would help the mRNA when it tries to exit the nucleus in order for it to go to the cytoplasm and go to the ribosomes. So stabilizes, yes, and facilitates the transport to the cytoplasm. Let me see. No question? Okay. The third process is called splicing. A cap, the 5 prime, a tail after we cut some here. The third one is called splicing. Firstly, before the splicing, we have to know that the mRNA, it has certain parts or certain segments that are coding, like the codes for protein, that are called exons. And I think you guys, you've been taught this, the exons, and some non-coding parts called introns. A way of thinking about it, I would think like exons, like expressional, is something that I would express from DNA to a protein, while introns, it could be like in between, like something that's non-beneficial, like non-coding in between, like introns. That's my way of thinking about it. You can use that if you want. So exons, like an expressional, it's a coding. This one is a coding, sorry. And the introns are not usually coding parts. Save this. Okay. So what is actually splicing? So we're gonna cut this out. Splicing means we're gonna get rid of the non-coding parts, like this intron here. Gonna cut it out and throw it away. And the cut is gonna be achieved by specific enzymes or a mixture of proteins and small nuclear RNA. Protein, small nuclear RNA will give us something called spliceosomes. The spliceosomes are the enzymes that are gonna cut the introns out, throw them away. Cut them out, then you basically join this end with this end, making a mature mRNA. Here you can see it. So you're gonna cut them out using enzymes called spliceosomes. What are the spliceosomes? Usually complexes of protein and small nuclear RNA. For you to cut both parts, you have to have the site, like the specific site for cutting. If you don't have one of these sites, it's not showing up, the sequence is different, the spliceosomes are not going to be able to cut in this part. You have to understand that. There is so the introns, when we cut them out and throw them away, they are cut in a specific way called lariat. A lariat is a specific structure. I'm going to show you in the next slide. So it's in a lariat form. Then we're going to throw it away. So this is another illustration or picture of what is actually happening in the splicing. So you have exons, introns, exon, intron, exon, and then the polytail. Gonna cut these out and throw them away. When you cut them out, it's gonna be in a lariat structure. What's the lariat structure? This is the lariat structure. You can see here. You can see this is what's called lariat structure. You guys can see it. So cut it in the lariat structure and throw it away. As we said, the introns are non-coding parts, and certain genes will have too many introns, like 99% of the gene are actually introns. And sometimes there would be like a mutation in these genes. An example of this gene is the dystrophin gene. This dystrophin gene will code for a protein called dystrophin. This protein is available in the muscles. So you have this protein in the muscles. And if there is mutation, usually the patient will have a muscle or a muscular dystrophy. That's a disease. Usually patients die in early age, like in teenage age. Some of the proteins, they just don't have any introns there. Therefore, they don't undergo the splicing, like histones. Histones are an exception in both the poly A tail and the splicing, the cutout. So histones, okay. Histones, they are an exception to poly A tail addition and the splicing. There's a concept that's called alternative splicing. So when you're splicing, you're cutting out the introns. Alternative splicing is cutting out at different sites, like each time you go. For example, usually we have exon, intron, exon, intron, exon, intron. We'll cut the intron like here and here and throw it away. The same will apply here and here, throw it away. But sometimes the cut would be here and here. So you will throw the, for example, the exon 3 with the intron 2 and intron 3, throw it away. That way you will have exon 1, you will have 2 and 4, because this is missing out. You already took this out. This is an example. This is an example, you can see it. Exon 1, 2, and 4, because we already cut here out and here. Sometimes the other way around, cut, for example, here and here. You're gonna throw the exon, the second exon, and you will have 1 and 3 and 4. The definition, basically, the alternative splicing is the process of selecting different combinations of splice sites, different combinations. Sometimes you got here and here instead of here and here as well. To produce a variable spliced mRNA. You would have like different forms of mRNA. These, the alternative splicing have different applications. Some of them are physiological, some of them are pathological. So alternative splicing, you don't cut or throughout cutting, you might miss some exons in the process. Okay, alternative splicing, there are two examples or two categories. One of them is physiological examples, and the other one is pathological diseases, basically. So physiological examples, one of them is the alpha tropomyosin example. This is a protein in the muscle. So we're gonna talk about that in the next few slides. Also some antibodies. Some of them are attached to a membrane of the cell, and some of them are secreted in the blood. Pathological, we have beta thalassemia, systemic lupus erythematosus, or for short, we call it SLE, or Hutchinson-Gilford progeria syndrome. Progeria means early aging. So basically, the patient is aging early while he's a child. Don't worry, we're going to talk about all of them. So let's start with the physiological, the alpha tropomyosin, and then the membrane-bound antibodies. So as a result of alternative splicing, you have the alpha tropomyosin gene. Usually there are 1 to 4 genes, alpha tropomyosin, like 1, 2, 3, and 4. So the alpha tropomyosin, the first one, can call it alpha, or the first one, the tropomyosin 1, usually have 15 exons and on chromosome 15. Due to different splicing of this protein, will have a huge variety of this protein in the muscle. Look at all the different, for example, here, we just cut this part, while in the above, we cut this here and here. So we got to the, also this exon in the middle. All of that is gone, for example, here, as you can see. So different parts, we cut in different parts. It's called alternative splicing. Just don't forget it. It's alternative splicing. Will give us different forms of the muscle or the mRNA and even different cells. This is another example. So as I said, from 1 to 4, we have 4 types. Sometimes you can call them alpha, beta, gamma, and delta. And due to alternative splicing, all of these forms are going to be produced. The second example is different antibodies. So we have the B lymphocyte cell, usually unstimulated. What does it mean, unstimulated? There is no antigen. This is just the normal thing that's happening in the cell. There are no antigens in the cell. There are no bacteria in the body. So the B lymphocyte cell will actually have the DNA, their DNA. This is, for example, the DNA. It's going to be transcribed. Look first at the DNA. There is a stopping codon here, and there is a second stopping codon here. What's going to happen in unstimulated B lymphocytes is that the transcription is going to be all the way until here. This is the normal, okay? All the way from here, starting from here, all the way to here, which is this example. You can see it. It's all the way to here. This is transcription, okay? The second stopping codon. So you have in this, and this is an exon. And this is intron. Here, all of this is intron. The spliceosomes will come and will cut this out. Basically, we're having a site here and a site here. Cut this out and reattach these parts. And in this form, we're going to have something called the IgM. This is an example of the IgM, or immunoglobulin M. So this is a form of antibody that's going to be attached to the cell. We're going to talk about how to differentiate between which one is which one with stimulated, which one without stimulation. Another scenario is that there's like a bacteria that's coming in the cell, in the body. B lymphocytes, they're now stimulated. So they will go through a shorter transcription here. They would go to the end of this transcription here after the stopping codon. So this is the end of the transcription. This is the example for it. We just went from here to here. We did not complete. We did not go to this side. We just have in this part that's small. So this is an example of the small. This is an intron here. Should be spliced out, right? But we don't have, the transcription ended before you have the part or the splicing site, as you can see here. Let me just erase some of these things that I. Okay. So this is where the transcription ends. And this is the splicing site. Remember at the beginning, I said there are no, for you to splice out, you need to have the splicing site. But here we ended the transcription before the splicing site. So you cannot cut it out. So what's the alternative? You're just gonna throw it away in the blood in order for it to go and fight these bacteria. So a way for me to think about it, unstimulated B cells or B lymphocytes will produce IgMs. The IgMs, remember the M goes with the M. It's a membrane bound. They're still attached. You can see it in this lecture. So this is IgM. The IgMs are attached to the cell. They're attached to the cell because they're unstimulated. There is no protein to go after. There is there is no antigen. Sorry, there is no antigen in the cell. There is no bacteria in the body in order for these antibodies to go and try to attack these viruses or antibodies. Yes, the antigen stimulated. If there is antigen, the B lymphocytes are going to secrete some of these antibodies. They're going to secrete antibodies in order to go for them and fight the antigens in the cell. These antibodies called IgDs. These are the ones. Number two. Code number 2. Number 1 here. Membrane bound IgMs. Just remember that. Membrane bound with unstimulated. No need to go after anything because there is no antigen in the body. So they will still be attached to the membrane. And because it's membrane M, you will have IgM here. The other one is going to be IgD. The second example of alternative splicing, the pathological sorry, yes, the pathological examples. First one is beta thalassemia. What's beta thalassemia? Is a problem with the hemoglobin protein. The hemoglobin protein have four components, four component proteins. Two are called alpha, and two are beta. So the problem in beta can be called beta thalassemia. What's the problem? This is an example of the pathological splicing. So basically, this is the gene, the normal gene of the beta globulin, okay? And what's going to happen is that you have these exons here, and these are introns. Exon, intron, exon, intron, and exon. There is a stopping codon here, okay, at this side. Usually it's going to happen, we're going to chop this off using the spliceosomes and combine all these exons together. That's what going to produce a normal beta globulin gene. But in a beta thalassemia, what's going to happen is that the splicing of the intron is going to be until this part, leaving the stopping codon here. So the stopping codon is going to be here. When you reattach all of these exons here, okay, when it goes to the ribosomes, start translation, protein, 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 there is a stopping codon. So the protein is actually will not be completed here. It's going to stop here because of the stopping codon. And thus you have a shortened form, or the short form of the beta globulin. This is pathological, and the hemoglobin is not going to function well. It's not going to carry oxygen to the tissues. The second one as well, you cut the intron here, but you left some part. There is some part that contains the stopping codon. When you rejoin, it's also going to happen. So B and C are pathological. The doctor also here mentioned that there are three segments. You have three segments here, but when it's long here, it contains a stopping codon. Also here, it has a stopping codon that will shorten the final protein after the translation. So thalassemia, alternative splicing, pathological example. The third one is autoimmune disease called SLE. Usually the patient will have autoantibodies that will go and attack. You remember when we said there is the alternative splicing, or the splicing of the, for example, let's say this is mRNA. We said we're going to splice the introns. Who is going to splice the introns? Spliceosomes. So if you have antibodies that are attacking the spliceosomes, that's going to splice these introns, so you're having a problem, and this is going to result in something called systemic lupus erythematosus. This is a very dangerous disease. Sometimes it involves the heart, the kidneys, the nervous system, and it can cause death. Usually it needs immunosuppression. The third pathological example of splicing is something called Hutchinson-Gilford progeria syndrome. Guys, any question? No question? I'm gonna ask question. Beta thalassemia. Is beta thalassemia a pathological or physiological example? Can someone answer me? Feel free to open the mic and answer, or write in the chat. Yes, beta thalassemia is pathological. Thank you, Saud. Yes. Okay, so Hutchinson-Gilford progeria syndrome is an aging syndrome. Basically, the child looks like an older person, and usually they die in their teenage ages, like 15 years old. They would die usually. What's the problem? there is a splicing problem here. This is a pathological examination. Usually, you would have a mutation in something called LMNA, or sometimes we call it lamin. Okay, you can see this is lamin A. So lamin A, or another name for it is LMNA, has different exons, and the problem is going to be a mutation in the exon 11. So the protein is called LMNA or lamin A in the 11th exon. What's going to happen? There's going to be a splicing. So with the intron that's here, part of the 11th exon is going to be deleted as well. So unfortunately, all of this from here is going to be deleted, including 50 bases deletion here. If you delete it, then you don't have a complete protein. And what's going to happen, usually the nuclear material, the DNA inside the cell, is going to be disorganized. There is no repair mechanisms, or they have defects in the repair mechanism. So repair of the DNA it's not good. The chromatin is disorganized. Basically, the nucleus is all messed up. And usually these patients, they would die early on. We call it de novo because it's a starting mutation. Why? Because it's not inherited. Why it's not inherited? Because these people usually, they don't live to reproduce. They don't live to get married and then produce like a children who have the same mutation. But de novo means from the beginning or the start. So Hutchinson-Gilford progeria syndrome, the exon number 11, problem in the protein or gene called LMNA or pre-lamin A protein. There is a deletion of 50 bases. It causes what? Nuclear damage, disorganization, and defective repair. You don't need to waste your time in this. Just memorize some of the pieces of information I just told you. The fourth process, remember we said there is a cap that we add, and there is a tail, and there is splicing, and we introduce you to something called alternative splicing. Now, sometimes when you have the mRNA, you will do some changes, or the body will do actually some changes. That's not splicing. We already talked about splicing. So any other changes other than splicing, we call it editing. So RNA editing is any change in the sequence of the bases other than the splicing. So let's read the definition. There are two definitions here. The first definition says the RNA editing is a molecular process through which some cells can make discrete changes to specific nucleotide sequence. So the change is actually in the sequence within the RNA after it has been generated. So you already did the transcription, then one of the processes is going to be the editing, changing of the sequence. Another definition says that these are changes to the RNA transcript sequence. After you already transcribed the DNA, you would have some changes. It says without splicing. Means splicing, we already talked about it. So any change other than the splicing, we call it editing. What's the function? Usually we'll have a large diversity or variance of the proteins, usually using just 22,000, 22,000 genes. Have only 22,000 genes, but like huge amount of proteins. How do you achieve this, or how does the body achieve this? The body will achieve this through using the processing and the editing as well. RNA editing. What are the types of RNA editing? There are 3 types, 2 categories. All of them, they include 3 types. The first category is called substitution. What do we do in substitution? So substitution will have 1 and 2. The first one is a substitution of A. Instead of A, we'll have I. What's that mean? Instead of inosine, which is a nucleoside, we will have adenosine. Sorry, instead of adenosine, we will have inosine. A to I. Instead of adenosine, we will have inosine. You can read a substitution of an inosine for adenosine. So we'll bring the inosine instead of the adenosine. A to I. What's the enzyme is gonna do that? It's called adenosine deaminase. How do you change the adenosine to inosine? Basically, get rid of the amine group. Deaminase, adenosine deaminase. But this one is specific to the RNA because there is another one in a purine salvage pathway that's different. So this one we have to specify that the enzyme that's going to change the adenosine to inosine as the adenosine deaminase that's acting on RNA, for short AR. Sorry. Note, the inosine usually will act like guanosine. A will be changed to I. Don't get mixed up. So A, adenosine, will be changed to inosine. How I think about it, I would have AI. I would think of AI and have like a small arrow here saying that the adenosine will be changed to inosine. AI, for me, the way I remember it, is AI. AI is a term that's now kind of well known because of the internet and the new. It's called artificial intelligence, الذكاء الاصطناعي. So artificial AI, the artificial intelligence, changing from A to I, from adenosine to inosine. The second one is changing the uridine to cytidine. The uridine nucleoside to a cytidine. So C to U. How do I remember it? CU. Like see you later, or like see you, from C to U. So I would think about the cytidine to uridine as see you. So see you later. This is like one benefit of saying to your friends, see you. That's just the way I think about it. So the C to U substitution, substitute the uridine for the cytidine using a cytidine deaminase, and focus on this one. It's a site specific. So there are certain parts of the body where you would have the cytidine deaminase, and certain parts you will not have the cytidine deaminase. An example of that, some proteins called the apoprotein. These proteins, the apoproteins, they usually help in the transportation of different types of lipids like cholesterol, chylomicrons, and low density lipoproteins. All these are going to be transported through the B100 and other forms also of apoproteins. So an example of that, there is an apoprotein. Okay, on to the next slide. So the apoproteins, usually they do exist in the liver and the intestine. They're like their gene in the cell in order for them to be produced. However, as we said, the cytidine deaminase is site specific. So it exists in the intestine, but not in the liver. It's in the intestine, but not in the liver. So what's gonna happen in the liver is that the apoprotein gene, apoprotein B gene, is going through this way because it's in the liver. Transcription, when it goes to the mRNA, there is no editing here because there is no enzyme. The enzyme is actually in the intestine. So you would have the apoprotein B100, which is full, complete, all of this gene transcribed from this side to this side. all of it. Okay, so full, it's a V100. Okay, however, in the intestine, you would have the enzyme, the cytidine, the cytidine deaminase. What's going to happen is that this codon, as you can see it here, the CAA. So this deaminase will deaminase the C, as you can remember, the CU, like see you later. The C is going to be changed to U, like from cytidine to uridine. So C here going to be changed into U, as you can see here. C is going to become U. UAA is a stopping codon. So when you go to translation, in order for you to produce the protein is going to be short, and it's going to be B48. So B48 is specific to the intestine, and it's not in the liver. So it's not in the liver because the cytidine deaminase exists in the intestine, so it gives the B48. As you can see here, I wrote down that it's in the intestine. It's going to produce, it's going to be responsible for production of apple B48 protein. So editing, this is an example of the second substitution, which is CU, C to U. The third one is insertion or deletion of uridine. So insertion or deletion of uridine, this is specific to uridine, and using an enzyme called the guide RNA, we call it for short the gRNA, which is also a site specific. So it acts on only specific sites. You would have the guide RNA that will introduce the uridine bases to a certain mRNA, or delete the uridine bases from certain RNA. As you can see here, there are multiple enzymes involved in this process. So what's going to happen is that this is the mRNA. We'll have the guide RNA will come here and attach to these sites in order just to keep the mRNA stable in place while the process happens. This is deleting the uridine. We have uridine. Here we don't have uridine, so we're going to insert some uridine here. So what's going to happen that the guide will come and attach. Then we will have an endonuclease that will come and cut. For example, here we want to delete these uridine, so we'll cut here and cut here and throw them away. Here we don't have, so we will cut open this mRNA and leave a space because we're going to add some uridine. The step after that is done by different enzymes, depends. If you want to delete here, you will use an enzyme called exouase. Exouase is going to remove the uridine bases. While if you want to introduce them, you have something called, you can see it here, the terminal uridyl transferase. So the transferase, the terminal uridyl transferase, is going to transfer or insert uridine bases into the mRNA, while the exouase is going to delete or throw away the uridine bases. Then we will ligate the mRNA using ligase. These enzymes you have to memorize them, guys. So the guide first, the nuclease will cut it open. You will throw away through the exouase, or you will add some uridine with the terminal uridyl transferases. Then you're gonna ligate it back. So this is the insertion or deletion of uridase using the site-specific guide RNA. Guys, what are the processes? You remember the processes or the RNA editing? What are the three types? We said there are three types: 2 substitution and 1 insertion or deletion. This is the insertion or deletion. This one is substitution. We came up with a mnemonic. There is a mnemonic for the first one. There is a mnemonic for the second one. What are the mnemonic for the first one? Guys, answer me in the chat, please. Said 2 substitutions and one insertion or deletion. Insertion and deletion is going to be for uridase. Yes, CU, this is the second one. The first one is AI, artificial intelligence. Yes, and since now we have Tesla and Elon Musk trying to come up with new artificial intelligence. So artificial intelligence, it seems like a common word now, which is the most common. I forgot to mention that. This is the yes, so it's the most common action. So AI, artificial intelligence now is the most common. You can think of it this way. So the most common is AI, or adenosine to inosine. CU, specific site, intestine. CU comes with intestine. Just remember that. Produces the apoproteins B48, specific to the intestine. It's not produced in the liver. So yes, so we already have the 4 steps right now. Don't worry, I have like a summary at the end. I'll show it to you. That was like kind of summarize everything. But like now, scientists are thinking about what's gonna what are the future of this editing. Can they use it toward humanity or not? So they're thinking about therapeutic. They might use it in some CNS disorders, central nervous system disorders, something like anxiety, depression, probably like increase the effectiveness of some medications. So they're going to use like 2 methods here, either to perform editing. There is natural editing like going on. We already mentioned some examples. See all of these examples: substitution, insertion, or deletion of uridate. All these are just happening normally in the body. So what the scientists are trying to do right now, either to perform editing, like artificial one, or using drugs to inhibit some of the natural editing that's happening right now, as the examples we mentioned for you. So either to perform editing or using some drugs to inhibit. So if they're going to perform some editing, especially in the CNS, what's the examples? One of the examples is something called 5-hydroxytryptamine receptor type C2. 5-hydroxytryptamine is a fancy name of saying serotonin. So 5-hydroxytryptamine is actually another name for serotonin. Serotonin is a neurotransmitter that's available in the brain, or also in different parts of the body. But now they're talking about the serotonin receptor that's in the brain. Specific category, it's called the C2. So 5-hydroxytryptamine C2 receptor in the brain. If they can introduce some editing to that, they might actually be able to like increase the effectiveness of certain drugs, or probably alleviate some of the CNS disorders, like anxiety disorders, like general anxiety disorder, or depression, or some of these. So 5-hydroxytryptamine C2 receptor, they can perform some editing on that. Or there is another receptor that's called potassium K, potassium. The V stands for voltage. So potassium voltage gated channel A. So there are multiple. There are A, B, C. We're talking about A, something called subfamily A. Sometimes they say A, or when they write for short, they use 1. And of this family, we're talking about the first number. It's called 1. So potassium voltage gated channel subfamily A, the first member, or member number 1. For short, we write it KV 1.1. 1 means family A. The 1 here means member, the first member. Or some drugs in order for to inhibit the natural editing, as I said. So therapeutic, memorize it. Just perform some editing or inhibit some of the natural editing that's happening using some drugs. what's going to happen if they perform editing, or which receptors. It talks about serotonin receptor in the brain, or the potassium voltage gated channel, subfamily A, member number 1. That's so all of what we talked about is actually in the eukaryotes. Eukaryotes like humans, but in the prokaryotes, it's a bit different, and there are not too many like differences. But you have to, I like summarize it for 3 points. Just focus with it. When you have here the mRNA, just focus with me here. mRNA, all of them are exons. Exons, all of them. There are no introns. Just understand, there are no introns here, for example. And when we say like this is an RNA, okay, this is all of it RNA. When it's in humans or eukaryotes, it's usually just one cistern. Cistern means gene. So all of this is just one gene. If it's in human, in eukaryotes, it's just one gene. It's not multiple genes. So the polymerase, the RNA polymerase 2, just will go for like each time we'll do just one gene. One gene. The one gene is gonna produce just one protein. You have to understand that. That's eukaryotes. While in prokaryotes can be multiple genes. Like this to this could be a gene. Like this gene. This is another gene here. They're connected, but like different gene. Or it could be just one gene in the prokaryotes. So there are no introns. It can be monocistronic or sometimes polycistronic, like multiple genes. There are multiple genes to be transcribed and to be translated as well. While in the humans, it's always monocistronic. It's one gene. Each time the polymerase will go, it's just one gene. Okay. So if the bacteria or the prokaryotes will have a polycistronic, how they're gonna go and translate that? We already said there are no introns in order for you to splice them. So how are you gonna go and produce like in the translation? As you can see here, these are multiple genes. This is a polycistronic prokaryote, and each time there is a beginning of the translation of the protein, and there is a stopping codon in one segment. Yes, for different gene, there's a beginning codon, there's a stopping codon, beginning codon, stopping codon. So when they go to the ribozymes, the ribosomes, sorry, outside the cell, each part, each gene is going to be translated as a different protein. As you can see here, translated independent, like as a separate protein. Like each one will be like, this one is protein according to the gene 1. This one is different protein. This is different protein. While in humans or eukaryotes, it's going to be just one gene going to be translated into one protein. Just remember that. How can they do that? Because each cistron or gene is translated separately. Humans, they only have one gene at a time, monocistronic. Prokaryotes, they can have mono or poly. And also another piece of information I forgot to mention, that the transcription is going to be by polymerase 2, usually for the messenger RNA. Polymerase 2 to produce the mRNA. And the second polymerase for mRNA, they don't proofread. They don't go back and repair like the DNA polymerase. Oh, thank you so much. I hope it was of benefit to you. Any questions? This is a nice summary. I'll recap. If you don't have any questions, I'll just go and recap. I don't know if I went too fast, too slow. I don't know. I just don't know how much of information you guys have, and your exam is tomorrow. So I tried my best to include not only the objectives, also the details in the lecture. Hey, you are welcome. So what we took here, mainly processing. So we said processing 4 steps. Adding the cap. Remember the cap, 7th medical guide, cap on the 5th end / the amine. Then we said adding a tail, a poly A tail in the 3, and there is an exception, there are histones. Then we did the splicing of introns, the non-coding part. There is also exception, histone proteins are an exception. Some examples or a concept behind the splicing, there is also alternative splicing. You have to know that. Physiological examples, alpha tropomyosin, troponin T also I did not mention, or the antibodies location. IgM goes with the membrane, and they're attached because there is no antigen to go after in the blood. So they will remain attached. This is the normal thing. There is an antigen, IgDs will be sent in order to attack the antigen. Pathological, beta thalassemia, SLE, remember, autoantibodies that will go and attack the spliceosomes. And Hutchinson, there is a deletion in the LMNA or the lamin A protein. There is a 50 bases that are deleted. And you can just use for short progeria, the early aging. Editing, there is substitution, AI, which is the most common. As you can see, AI goes from adenosine to inosine, or CU from cytidine to uridine. An example of that, the ApoB100 gene editing. And in the intestine, there is V48 version of the protein. There is V48 and 100, while in the liver there is only 100. Adding or deleting the uridine using the guide RNA. Uridine, you add or delete that using the guide, and some other also enzymes involved. You have to memorize it. Therapeutic, 2, perform editing in 2 receptors. Serotonin receptor in the brain to help with the anxiety and some other disorders, or the potassium voltage gated channel subfamily A, number 1, or using some drugs to inhibit the already existing natural editing. Prokaryotes, no introns, monocystronic, sometimes polycystronic. How they can deal with polycystronic, they translate them as separate proteins. And hopefully I did not take too much time. If you guys want me to go throughout the lecture, yes, sure. The guiding RNA, sorry. Let me just delete. So basically what's going to happen, okay, Dania, I'll explain now the insertion or deletion using the guide RNA. So the RNA is this one. It's going to come and attach, because we're going to cut the mRNA open 2 parts. They might degrade, go and degrade, because we're going to cut it out. So you have it here. It's going to attach into the mRNA to stabilize it. So as they call it, the hybridization of the RNA with the guide RNA. So the guide RNA will actually assist and try to stabilize. Then another enzyme called endoribonuclease, or endonuclease is another name for it. It's gonna come. It depends if you wanna add or delete. It's different. So if you wanna delete, gonna cut on both sides here. The function of the endonuclease is just to cut. It's always just to cut. They're not gonna do more than that. Just cut. They cut it here. Then what's gonna remove here is exouase. Exouase is gonna remove the uridine nucleosides from here. Gonna remove them. If you wanna add, the addition is gonna be different by something called terminal uridyl transferase. Just think of it as transferase. It's gonna transfer the uridine into the mRNA. Exo, like out, it's gonna delete the uridine bases. So exo, out, the uridine bases are out. We're gonna delete them. they're going to transfer them into the mRNA here. When you transfer them here, for example, also you want something to connect what the endonuclease is already done. They cut here before we add the nucleases. You want something to ligate here, or it ligates. So endonucleases, they always cut. Ligase, they bring together and reattach something that's already cut. So you bring together and attach again. Come here. The guide RNA is going to guide the process by stabilizing the messenger RNA. Endonuclease is going to cut. If you want to add, you will use the terminal uridase transferase to add the uridine bases. If you want to delete, you will use the expo UAs. It's going to delete some of the bases. Ligate or ligation is going to be done by the ligase. Ligase will go and attach. And you already have the changes. Here was uridine, we got rid of it. Look, there is no uridine here. Here there was no uridine, we cut it open, then we added uridine and reattached the sides. I hope that's clear. If you want me to repeat that again, I can go ahead and repeat. Thank you, guys. If you want me to go through the original slides of the doctor, I have them with me. I can go through them, whatever suits you guys. If you want to take a screenshot of this one, go ahead. I think this one like summarizes most of what's in the slide. Only here in the guide RNA, we did not include some of the enzymes. Any question? You guys are more than welcome. Okay, خلاص.